My name is Krzysztof Palczewski, and I am faculty in the Department of Ophthalmology, Physiology and Biophysics, Chemistry, Molecular Biology, and Biochemistry. So my interest is in biochemical processes of our vision, and I arrived here to Irvine about 7 years ago, and I'm part of the Center for Translational Vision Research. My lab is interested in two fundamental processes that initiate our vision, and they are called phototransduction and visual cycle. So phototransduction is the step which enable light stimuli to be translated into a biochemical chain of events in the rod and cone photoreceptor cells that ultimately send electrical signal to the brain. But it would not be sufficient to sustain our vision. That process has to be regenerated continuously, and that is enabled by metabolic pathway called visual cycle that produces the light-sensitive chromophore 11-cis retinal. The proteins involved in this process need to be properly folded and properly interacting with other partners in the signal transduction. Therefore, any mutation in those genes will lead to retina disease. The question arises, what can we do to correct those mutations? What we can do to prevent developing retinal diseases? And there are two aspects that one could take in order to alleviate the problems related to the mutation. One is a very simple classical pharmacology, which we call systems pharmacology, that we were able to stabilize photoreceptors that can function for a little bit longer. A very recent contribution to modern pharmacology is the gene editing and gene augmentation, in general, gene therapy methodology that enable us to either deliver the mutated gene, inactivate or inhibit the mutated gene that has now pathogenic property, or we can go directly to the DNA and to make modification of the DNA to rescue the normal function. So overall, there are a lot of genetic approaches that could lead to improvement in the outcome of these inherited diseases. Most elegant are those changes that we can change the mutant form of the protein into a wild-type protein. And in this case, it will not be a treatment, but rather will be a cure, will be complete reversal of the property of mutant protein back to the wild type, and no need for therapies that need to be followed throughout the entire life. When rhodopsin was discovered, it was discovered by one fellow who was able to isolate rhodopsin and rod cells from frog by himself and do all of those experiments individually. This approach today is impossible because of the complexity of technologies that we need to bring into understanding as well as treatment of those eye-related diseases. Therefore, it is critical that we accumulate expertise, we collect the expertise in one place, like CTVR, Center for Translational Vision Research, that will enable us to have that expertise in-house, that each of the investigators and their team will be able to add value to overall approach and understanding what will be required next from physical biochemical processes to pharmacology to regulatory and also will also keep eye on very important ethical issue related to genome editing that will require us as a society to think through what it's allowable and what shouldn't be allowable to modify our genome. The centers like ours, if it is based on interest and excitement, what needs to be done next with proper background and expertise, can bring revolutionary changes. And I hope that our center will be one that will lead those changes in vision research, not only here in Irvine, not only here in California, but in the world.